Hi everyone, this is the fifth mini lecture for chapter 13, and in this one I have the learning outcomes placed in this slide. We're going to be looking at B cell activation and starting to talk about what antibodies are, not only what their structure is, but the function of the different types of antibodies. So this is table 13.6, and just like I said about table 13.5 on T cell activation, this is a really good summary for B cell activation and would be a great place to really make sure that you understand. So the interesting thing about B cells is there are two ways that they can be activated. The first is pretty simple, and that's that they actually bind that microbe or antigen, and that actually activates them. The second way that they can be activated is that they are actually activated by a T helper cell that helps recognize and helps activate the B cell by recognizing that antigen. And so that would be where we would have B cell and T helper cell cooperation and recognition. But in either case, whether it's through the T cell or that it's just the direct binding of that particular microbe or antigen, then we have an activated an activated B cell. And an activated B cell has a number of different pathways that it can start to follow. The first one, of course, is that we would have the memory cells. That's one of the ones that we saw with T cells too. So it will proliferate and have memory cells that have that specific antibody that would recognize that antigen in case we have a second exposure to the particular antigen. It can also produce regulatory B cells, and these have the ability to secrete interleukin-10, which regulates T cell response. And then one of the more important things is that it becomes plasma cells. It can make plasma cells, which then can produce antibodies that are specific for the particular antigen that it was exposed to up in step 1 or step 2. And it's these antibodies that actually play the big role. And so when we look at antibodies, we want to talk about what their structure and function is. They're a symmetrical Y-shaped arrangement, and you saw that in one of our earlier slides. They have an actual portion that is the antigen binding fragment. It's called the FABs, and this is the Y portion, that forked portion of the Y. And it has a groove that can accommodate whatever the unique epitope that one is unique for. It has these identical heavy chains that make up the lower part of the Y, and the identical light chains, and these make up the lower part also. I'll show you a picture of that. So we see here that we have that FC region, which is considered the heavy chains that are going to make up the major portion of the Y. We have the light chains, which are these outer portions of the Y. The V stands for variable. This is where we see all of those different amino acid sequences that can make that particular antigen binding site unique for any given antigen. And then this is the bottom portion of the antibody, and it is the binding site for cells. So where it is bound to a B cell, for example, sticking out to grab onto that antibody. And this is an actual picture of what one looks like. So that antibody-antigen interaction that we see at that hypervariable region, this is where that epitope would bind to the antibody. And remember that the amino acid content of that region is varied so that we have a specific antibody region, hypervariable region that would recognize any given epitope. So it's going to be very much like enzymes and substrates. Remember when we talked about kind of that lock and key, only the particular epitope will be able to bind the antigen specific for it. And both of those Fab sites, so if you go back and you look at the two Fab sites here, these are identical. So that immunoglobulin or antibody molecule can bind the epitope on the same cell or on two separate cells. So it might actually be able to bind one here from one cell and one here from another cell. And what we see with that is once those antibodies then are produced by our plasma cells, what do they do? So what can we do when we release these antibodies? There are a number of different things that can happen. The first one is that it can actually coat the surface of a bacterial cell. So it's binding to some epitope that is being shown on that bacterial cell, and this can maybe stop the function or replication of the bacterial cell. We can also get something that is called opsonization, and what this is that by binding onto these antigens on the bacterial cell, these antibodies make it easier for a phagocytic cell to come along and phagocytize it. So that's a way of making it easier to grab onto and to engulf. Another thing that we can have, and this is common with viruses, is we can have neutralization. And what that means is that these antibodies will actually block the binding site on viruses, and they can't bind the host cell. So it prevents it from attaching, which means it can't infect. Another thing that we can get is something called agglutinization. This is where antibodies will bind more than one. Remember, they have those two variable regions, so they might bind this bacteria and this bacteria. And if we get them all clumped together, generally speaking, they're not good at mobilizing them. They can't move, which makes it easier for phagocytosis to occur. And then another one, we talked about the complement system, the complement pathway in the last chapter. If we get these antibodies, this often allows for binding of the complement proteins, which can puncture the bacterial cell. And then the last thing that can happen is sometimes it can work as an antitoxin. When a toxin is produced by a bacteria, the antibodies might be specific for that and actually bind and stop them from attacking the host. So there's also an important function of the consistent, the region that binds the cells. So the Fc region can actually bind the membrane of macrophages. So once the variable section has bound onto an antigen, then it can take that bottom portion of the Y and bind to a macrophage. And in that sense, it can actually make it easier for phagocytosis to occur. So again, we talked also about opsonization, which is the attachment of that antibody to those foreign cells, and then that Fc portion will attach to the phagocytes, which allows the phagocytes to get a good grip and actually engulf the antibodies and the invading cells. We have different classes of immunoglobulins, so different classes of these antibodies. They're considered isotypes, which means that there are structural and functional classes within the whole overarching term of immunoglobulins. So we can have IgGs, and these are monomers that are produced by plasma cells. And they're the primary response and memory cells in that secondary response. So they come from the plasma cells in a primary response, and they come from the memory cells when we have a second infection, perhaps, by a particular bacteria. IgGs are the most prevalent antibody. Most of our antibodies are IgG, and they have the ability to neutralize toxins, to do opsonization, and also to help fix complement, so to help those complement proteins attach and puncture a particular foreign agent. The next one would be IgA, and these are the monomers or dimers, meaning a single antibody or two together. The monomer we see in small quantities in the blood, and the dimer we see in mucus and serous secretions of different parts of our body. We have IgM, and it's a pentamer, which means that there's 5 antibodies that are all bound at the bottom portion of the Y, and they can do a first response to an antigen, so they can serve as that B cell receptor. We have IgD, which is a monomer, so a single Y-shaped immunoglobulin that can act as a receptor on B cells. So it's the one that would stick out on those B cells and recognize an antigen, and then when it recognizes one, it would activate the B cell. And then the last one is the IgE monomer. These are antibodies that we see that play a role in allergy, worm infections, and some other things that we see with allergies, anaphylaxis and asthma, for example. And there's a nice table in your book that actually shows what these each look like. So take a minute and look at those. And then the last thing is to think about, we've talked about specificity and that antigen to antibody, but the second thing we discuss with this specific immunity is the idea of memory and why that's important. And this is a good example showing the primary and secondary response to a given antigen. So this is showing on the y-axis, we're seeing antibody titer, meaning the amount of antibodies present in the blood. And along the bottom is time, how long something would take. In orange over here, we have a primary response. So we'd have that first exposure to an antigen. We have a very long latent period while we're waiting for it to be recognized and to activate and stimulate B cells, T cells, all of that. And then we see this increase of different immunoglobulins, so different antibody reaction. And it's showing at the highest peak here. Now, if we were to go for some variable time interval, this could be weeks, months, even years, we have that second exposure to the original antigen. And what we see is no latent period. We get this immediate reaction of the different antibodies. And that not only happens much more quickly, but at a much higher rate, which means that the infection is taken care of much more quickly with the second response. So when we check in here, we need to talk about activated B cells, what happens, what are the possible outcomes, the memory cells, the regulatory, and the plasma cells. The different ways that antibodies can work, make sure that you know the vocabulary words. And then also looking at the immunoglobulins, the different types and where they would be found and what they might do. And then last, being able to differentiate the primary versus secondary response to an antigen and why that's so important in this third line of immunity. So that's the end of 13.5 mini lecture, and we have one more mini lecture for this chapter. Hope this is helping.